Well, it's so wonderful to see you all tonight. A very warm welcome to our first resident member series of the 2024-2025 academic year. Welcome. We'd like to begin our time by situating ourselves in the land that we're on, located on the traditional, ancestral, and unceded territories of the Hunkaminam-speaking Musqueam people, and recognizing that this land has always been a place of learning, and we like to always begin and really ground ourselves and situate ourselves on this space, this beautiful land that we call a place where we've kind of decided to be at Green College. We're really delighted to be able to host these resident member series every single Monday with you all. Thank you for showing up and thank you for being here. We're really treated to have Erin speak on her research tonight with us. Those who remember Erin's previous talk last year, it was absolutely fantastic. And so we're excited for what you have in store for us tonight. Just a little bio on what Erin's research is about. Erin Evans is a PhD student in the Department of Microbiology and Immunology here at UBC. She was born in the UK and grew up in New Zealand. Her work focuses on ancient viruses embedded in the human genome and their possible role in causing MS. Outside of the lab, she's a swim coach and champions neurodiversity education, showing that society has room for and thrives with all neurotypes. With that, I'd like to open the floor up for you, Evan, and then we will then proceed with question and response afterwards. Amazing. Please welcome Erin Evan.
Is everyone able to hear me okay? Cool. All right. Perfect. So what I'm going to talk to you about today is what we know and what we don't yet know about multiple sclerosis. So this is related to my PhD in microbiology and immunology that I do here at UBC. And I'm really excited to be presenting here to you guys today. Okay, so a little agenda for the evening. So first I'll give a quick explanation of what multiple sclerosis is, and then some ideas about the causes or risk factors of multiple sclerosis, going specifically into Epstein-Barr virus, vitamin D, and sort of like the genetics aspect of it. And then ending on some treatments for MS and sort of some exciting research and where the field is going. Okay, so a little bit about me. I'm a PhD student in microbiology and immunology at UBC. I'm in the labs of Drs. Maria Tokiyama and Mark Horwitz. And I study the relationship between ancient viruses that are embedded within our human genome. So they're part of our genetics, they're part of who we are. And how this can relate to the onset of multiple sclerosis. So I'll give a little bit more of an explanation towards that at the end. But I thought it was important to give you a little this is what I do in the research, and then give you an idea of actually what is multiple sclerosis. And why am I interested in it? So my dad has type 1 diabetes, which is an autoimmune disease, and my grandmother has multiple sclerosis. So I've grown up kind of in this environment of autoimmune diseases around the place. And from a young age, I was just quite curious about why this was happening. And so, right. What is multiple sclerosis? It is a chronic autoimmune demyelinating disease. And I understand that's a lot of words, so we're going to go through them. So chronic means that it causes a lifelong disability. So the average age of diagnosis of multiple sclerosis is between 20 and 50. So it can end up having quite long-term effects for the individual. The term autoimmune means that it's where the immune system, which is normally there to protect us against outside pathogens, is when it starts to attack our normal body tissues. And then demyelinating is damage to the insulating layer that surrounds our brain cells. I'll explain a little bit more about that in a bit, but essentially multiple sclerosis is a disease of the central nervous system. So on the image here, we have the brain stem and then the spinal cord. And then this is a sort of figure by Robert Carswell in 1838, and he was marking out MS lesions. So areas of demyelination that we can see. In this, it was from an autopsy, but now we can see it in non-invasive methods, thankfully. So types of symptoms. Honestly, wide range of symptoms involving all the organs that you have in your body, basically. So we've got personality and mood changes, fatigue, numbness in various limbs, and then it can affect different muscles. There's an increased risk of osteoporosis, and it affects muscles, not just the arm ones and the leg ones, but also the ones that control your bladder or other parts of your body. And there are 3 types. We have relapsing remitting, which is in the blue, which is this idea where you have attacks. So you, as time goes on, you have an attack, but then you recover, and then you go a period of time where you don't have any symptoms at all. And then there are some new symptoms or attack, and then a little bit of time, and then attack. But over time, it's gradually getting worse because you're never being able to recover completely to the level that you were before you got it. Sometimes relapsing remitting MS can turn into secondary progressive. And that's where you initially have these relapses and then it turns into a chronic form. And I will say that the majority of people with MS have this relapsing remitting. And then a portion of people will go on to get this secondary progressive type. And then finally, the rarest type is primary progressive, where from the outset, there's no relapse. So I want you to, we're going to talk about the immune system specifically in the brain. And I understand that the immune system can sometimes be a bit terrifying because there's all these cell types and we don't know what to do. One sec, I'm going to admit my sister.
I've got you, Erin.
She's joining. Amazing. So imagine that we have the brain in grey. No, the brain is in grey. And then we have not brain. Now I understand that the body is not just brain and not brain. For this circumstance, it is. And the brain is surrounded by this blood-brain barrier, which protects it from lots of different things. Because our brain is very important, and we need to protect it from all the external pathogens and everything that is out there. We need to protect it. And for a reason that we don't quite understand yet, the blood-brain barrier starts to break down in MS. So it starts to become permeable. And what happens when it becomes permeable is that we have different immune cells trying to enter the brain. So there's a whole host of them, but the ones I'm going to focus on are called B cells and T cells. I'll explain a little bit more what they do later on. But essentially, they are directed towards the brain, again, for something we don't fully understand. But then we get all these immune cells in our brain, and we don't normally have them in there, and we don't really want them in there. Now, this is a neuron. So this is basically what helps to send all the information in your brain from your brain to your limbs. Around the edge, it has this insulating layer, or the myelin sheath. If you imagine a wire, we insulate them so it runs faster. That's exactly the same as neurons. But what happens when the immune cells infiltrate into the brain is the B cells start to produce these things called antibodies, which are targeted against this myelin sheath, and it starts to break it down. Because normally these antibodies are directed against pathogens, and that's a good thing because they latch on and it tells the body, we want to kill this, it doesn't belong here. But instead what is happening in this case is that the antibodies are attaching to the wrong thing and still saying that we need to kill this, even though it's a healthy tissue that you need and that you want. T cells, on the other hand, like to come in, that's another person, like to come in and produce lots of cytokines. So these cytokines are basically little molecules, and they like to cause havoc wherever they go. And they like to attack things and destroy things and break things down. And these T cells produce a lot of these cytokines. And what this leads to is that the myelin sheath is broken down. And so it can't work as well. And impulses from your brain They go through your neurons, but they're going a lot slower than they would normally. And so it is a lot slower to get to your body's muscles, and basically everything that you need in your body is controlled by nerves. And so that's the general immunological basis of multiple sclerosis and why it's causing these symptoms. Okay, so even though I've just told you what it seems like, oh, we must know everything about multiple sclerosis, there's nothing left to learn. There's a lot. So multiple sclerosis is the most common autoimmune disorder of the central nervous system. About 2.9 people have MS globally, and it is the most frequent cause of non-traumatic neurological disability in young adults. And I'll explain the end bit there in a bit. And so current research in MS is looking into causes and treatments, but also understanding the different presentations of multiple sclerosis. So relapsing remitting, I've said, is the most common type. And so because of that, we understand relapsing remitting MS a lot more than we do progressive MS. And so a lot of our research is trying to go into learning about progressive and why it is a lot more severe and quicker than relapsing remitting. We're also looking at biomarkers. So one of the ways that we diagnose MS is we do an MRI, which is fine, but people aren't getting MRIs on a regular basis. And so it might be a while before that person who is experiencing these kind of generic and vague symptoms, could be a while before they actually get an MRI of their brain. And so if we look at biomarkers, that is a much quicker and easier way to identify MS in somebody. And as we all know, if we have earlier diagnosis, we have better treatments and cures. We're also looking at improved imaging. So I said we use MRI, and there's always ways to improve it and get better images that are more meaningful to us. And then finally, understanding how disease progresses. Because we know a lot about the immunology, but we don't know everything, unfortunately. Okay, so now I'm going to go into a little bit of what we do know about MS and how we can use that to kind of look at susceptible populations. And so through this, we can use something that's called a genome-wide association study, or GWAS for short. And what we do is we get a population of people with MS and a population of people without MS, and we compare their genomes. So we look at their different genes, and we look at specific tiny, tiny differences in those genes. As in, a gene could be this big, and we're looking at a tiny centimeter of a difference. And what this means is it can give us a genetic susceptibility. And it's displayed in this plot, which is called a Manhattan plot. And it's called that because it looks like the skyline of Manhattan. And the higher ones are the ones that are more different in people with MS than those without MS. So this one at the top, we can see it's pretty big significance. And that's something that's called MHC. What is MHC, you say? So MHC stands for Major Histocompatibility Complex, which is, again, a lovely long name that scientists like to give things. But essentially, it goes into how we recognize pathogens. Unfortunately, the body can't just go, oh, this is a pathogen. They don't have a flag that they're waving to say, oh, hi, look out, like I'm trying to do you harm. What happens is that these pathogens infect cells, and then these cells express proteins on the surface. And a virus comes in and is gobbled up by a cell because the cell thinks, oh, that could be a nutrient, discovers it's not a nutrient. So decides to display it on the surface of the cell using a protein, which is the MHC protein. And what this does is this creates a little flag for the immune system. Our T cell comes along, recognizes it, and then our T cell can actually go out there and fight off the pathogens. And these proteins here, so this one sort of expressing the little pathogen, differ slightly between people. So it's one of these things that's like genetics, like you pass on your hair color, you would have also got your MHC class from your parents. And we have some versions, which we call haplotypes. They're more common in people with MS than those without. And so the one that we have called the lovely name of HLA-DR2, which basically means that of all the options you've got, say it's a rainbow, that's the orange one. And this was done looking at thousands and thousands of people with MS and their close relatives without MS. So that's the genetics. That's the part you can't change. You can't change the genetics, I'm afraid, even if the media will think that you can. But we're going to look into environmental risk factors, and actually some of them you can change and some of them you can't. It's really on this timeline that starts even before you're born. So even the environment that your mum is in when she is pregnant with you can affect your susceptibility to multiple sclerosis. So on the bottom here, we've got vitamin D. So vitamin D we can get from the sun, but we could also get from supplements. And this is really important between when you're in the utero and up to adolescence. Smoking is also a really massive risk factor for multiple sclerosis, which is entirely preventable, unless it's the passive smoking when you're a child. Obviously, that's a bit more difficult. When we think about, we've all heard about the microbiome, which is the population of bacteria that live in your gut and seem to influence everything from the way you think to what type of exercise you like. And we know that changes in the microbiome can lead you to being more or less susceptible to multiple sclerosis. Here we have Epstein-Barr virus, and I will go into that way more in a little bit because that's one of my favorite risk factors, I guess, of multiple sclerosis. And then at the top, we have stress. We all know it's not good for you, and it's not as if you're really stressed for even 10 years, that's not a guarantee that you'll get multiple sclerosis. It all has to come together with your genetics and the environmental risk factors to actually produce a risk of you getting multiple sclerosis. So I'm going to focus on vitamin D and Epstein-Barr virus. So vitamin D. So if we look at the prevalence of multiple sclerosis and we look at the countries with the highest prevalence, we notice that they are in the northern hemisphere or in the bottom of the southern hemisphere. So we have this sort of like almost a line in the middle where there's very low rates of multiple sclerosis. And Canada, being quite a northern country, has quite a few. And this incidence of 250 individuals per 100,000, that equates to about 90,000 Canadians living with multiple sclerosis. And so when they were looking at the prevalence of MS and they saw that it kind of looked like this on the map, people were thinking, well, what's that got to do with it? People would think, oh, it's because those countries are the richer countries, and maybe it's this hygiene hypothesis, which is this idea that as we're getting cleaner, we increase our susceptibility to autoimmune diseases. And that may also play a role. I'm not going to go through that today. But there were people who looked into levels of vitamin D. And so they looked at people who had immigrated from India, Pakistan, or Bangladesh to the UK before and after the age of 15. And what they found is that if you were under the age of 15, when you immigrated to the UK, you retained the susceptibility of MS of India, Pakistan, and Bangladesh, which is lower because they have more sunlight, vitamin D. And so their rate is about 1.33 people per 100,000. When they looked at people who had immigrated after the age of 15, they found that they gained the risk of people who lived in the UK. And this risk is quite large, 190 people per 100,000. And so what they determined from this is it's that your vitamin D exposure prior to the age of 15 is really vital in your MS susceptibility. So in places like Canada and the UK, the northern parts of America, and down in the southern hemisphere, parts of New Zealand, we have much higher rates of multiple sclerosis in comparison to those at the equator, simply because we get less vitamin D. For 9 months of the year in these countries, we do not get significant levels of vitamin D from the sun, which is why it's important to take, sorry, I saw a question on there, and I think you can see it on there, yeah. Sorry, I will answer questions at the end. That's why it's important to take supplements, but it's particularly important for young people living in susceptible countries. So I think that's quite striking effect. Okay, so my favorite, Epstein-Barr virus. So does anyone here know that they've been infected with the EBV, or maybe were diagnosed with infectious mononucleosis, kissing disease, glandular fever? Up your hand. So I reckon it's probably more than you, because 95% of the population are seropositive, which means you've been infected with EBV and mounted immune response against it. It typically occurs between childhood and adolescence. If you're a child when it happens, it's usually asymptomatic, so you wouldn't know. And if it happens when you're older, perhaps when you're sharing saliva or kissing other people, that's when you can get it. And that's when it might present itself as infectious mononucleosis, where you have fatigue, general flu-like symptoms, but then you also might have some swelling of the lymph nodes and tonsils, and your spleen enlarges. Basically, you're just quite ill. And this is a little microscope picture of EBV. Okay, and so there was some knowledge about, it seems that people who had EBV, particularly those who got infectious mononucleosis, seemed to have MS. And so what this paper did is they got 10 million military personnel, which is a lot of people. And the thing about the military is that they will give their blood because they're told to. And then we, as scientists, they're told to because they have tests done. And then those tests are measured and they're stored. And what scientists can do is they can take those results and actually use them to do massive studies on an amount of people that you wouldn't be able to get normally from a population. And out of these 10 million, they found 955 people with MS. The next one is going to be wrong. That should say 954 people were previously infected with EBV. I'll explain the one person in a second. And so what they did is they took people who had all been seronegative at the time of their first sample. So they've entered the military, they're 18 years old, the sergeants are like, give me your blood, and the guy goes, okay, yes. They're all seronegative, which means they haven't been infected with EBV. And then they followed them until they did get infected with EBV. And so, as I said, 95% of the population do get eventually infected. So obviously there's quite a high number of those people got EBV. But when we compare people with MS and people without MS, we can see that the people with an MS were almost 100% seropositive compared to the individuals that were kind of around the general population, which was around 50%. And this is, it might seem small or unexciting, but in science, where nothing is certain, that's pretty certain. And it's pretty cool because it means that we can investigate it further. So you might be thinking, so what we kind of determined from that paper and other papers and studies that have been done is that infection with EBV is required, but not sufficient for the development of MS. As we know, like 95% of people have EBV, not all of them are going to develop MS. And what about that one person who didn't seroconvert? Well, maybe that they seroconverted later, between the final serum sample and the development of MS. It could be that they're just a weirdo. It could be that the test was wrong. There could be many things, but the fact that it's only one person out of nearly 1,000 is pretty damning evidence. Okay, so I've told you all about MS and how bad of a disease it is. So how do we treat it? So if we go back to the 1990s or even before, when my gran was diagnosed, they gave her a massive dose of steroids and then told her to move into a bungalow and just live out the rest of her life, knowing that she's saying, you'll be in a wheelchair, you won't be able to cope, like, you know, not give up on your life, but this is all we've got, steroids, which if anyone's been on steroids will know that they're not fun. Since then, thankfully, we've gained much better treatments. So it started off with these interferons, which are molecules that are present in your body anyway, and it just gives you an extra boost and sort of tells your immune system to stop attacking things. There's a side effect of this, that it generally makes your immune system less active, so you're more susceptible to various illnesses. But that is easier to deal with than the symptoms of MS. More recently, we've been getting these things that have these funny names that end in MAB. So that just means it's an antibody. So it's something that we've developed to target certain areas of the immune system. So it might target those T cells that we talked about, or target those B cells. And what it can do is it can redirect them, or it can dampen them down. So they're sort of like, I'm active, I want to fight everything. And then this antibody just comes around and says, calm down, please. And so there's a lot more treatments out there, and these can be used for different types of MS at different stages of severity. And so that's good. And finally, I wanted to go into sort of what are people researching in MS. So this summer, I was lucky enough to go to a summer school where I got to talk to lots of other people, lots of other researchers who are researching multiple sclerosis. And I got to see outside of my field, like in my lab, we're very focused on multiple sclerosis and EBV and our experiments. And this opened my eyes to what other researchers in my field are doing. So some people are looking at improving MRI techniques. Other people are looking at developing vaccines against EBV. So if we know that EBV is required, if we prevent it even happening in the first place, that might help prevent cases of multiple sclerosis later on. And then finally, something I look into is reactivation of EBV by various factors. So for example, these endogenous retroviruses that are part of your DNA might like to hop out occasionally and activate parts of EBV in your body that have sort of been lying dormant. And then that might end up activating the immune system towards attacking your brain and developing MS. Obviously, there's a lot more outside of this. This is just a little snapshot of what's happening. And with that, I'd like to thank you for listening and ask if you have any questions. Yeah, I can monitor. Great.
Actually, I think it might be able to pick up from the big microphone.
Shall I just speak, and if you can hear me, confirm?
Ooh.
Okay, let's use mine. It's fun anyway.
Okay. By the way, that presentation was incredible. The graphics were just wonderful, and it's often quite rare to come across a presentation where you can give an entry-level thing, and it's just like, oh, I understand. It's amazing. So thank you. My question actually was a little bit off-piste. I know you study Epstein-Barr virus in particular, but at the start of the presentation, you mentioned the blood-brain barrier and the fact that that breaks down, which is one of the major things that causes the infiltration of all of these immune cells. Is it possible to is any research looking at how we can restore the blood-brain barrier? And is that one avenue of research that is interesting or worth note?
I imagine there is. I'm not aware off the top of my brain. I think there are also drugs that are trying to prevent immune cells from crossing that blood-brain barrier in the first place. Because there are some ideas that actually there's a few immune cells that get in there first and then they start to break down the blood-brain barrier. So if you prevent that from the onset, you can prevent downstream. But no, good question. I like that.
Thank you.
Thank you, Erin. As somebody who's lived with MS vicariously, my mother first developed some of her earliest symptoms when when she was carrying me. It's been difficult watching her with this progressive condition. I'd be interested, especially with you sharing a bit about your grandmother. And again, I don't know if this is what you really researched, but I'm guessing something you might know a little bit about. What are some of the ways that people that are dealing with disease now 30, 40 years on, getting, at least in my mother's case, she's getting towards the end of her life. Are there ways that we're able to still provide supportive care now that a lot of what's been done is done? They can't really, as I understand, they can't really repair the lesions, and they're creating, at least in my mother's case, kind of secondary and tertiary parts of what makes her life interesting in some sense, but difficult in others. Anyways, your thoughts would be really appreciated, even if it might be ways that I can understand her better and support her.
No, it's definitely, once lesions are created, it is really hard for us to mend that because we still don't understand the processes underlying repair that happens. So we kind of have different avenues that we can go with treating individuals. So we can go with these sort of what I explained, the disease modifying therapies, which is where we use antibodies or other chemicals to try and redirect the immune system somewhere. We also have symptom management. So whether that's through drugs to relieve pain or make muscle relaxants to kind of like ease stiffness. We can also go down that method, and they can be done in tangent. And then finally, a big one is physical therapy, is making sure that people are able to still continue to move their body and engage their brain and their muscles. And this was something that actually my gran was really, basically when she got told to move into her bungalow, she went, no, I'm not going to do that, because she's very stubborn. And she instead said, no, I want to go to physical therapy. And so she started to go to physical therapy, and then gradually she had an attack where it kind of paralyzed her. And then through exercise, and I'm sure other treatment, she was able to regain some more movement, which obviously was, you know, very helpful for her long-term and short-term, looking after 3 young children. So there are, I think people are looking, when we look at the basic science route, which is a bit more cold-hearted, and then there are also people looking into the clinical side of it and trying to make people's lives better now. So yeah, thank you for sharing about your mum.
Thank you so much, Erin, for the great presentation. I'm just curious, so for the slide where they did the study with 10 million military personnel, I was curious if they controlled for different biases like gender or like race and that kind of thing in the study, just because I know like stereotypically the military is a lot of like white men. Or men in general, I guess. And yeah, I was wondering if they controlled that.
No, that is definitely a big consideration, is that the military is not a particularly diverse set of people. And it is obviously, it was the US military, so it is very defined into the US population. That being said, they matched their controls very well. So they matched each person with MS to 2 or 3 controls. And when I say matched, for example, you and me, Alex, if we were in a study and I was the diseased participant and you were the healthy participant, we would not be matched up because we're not the same sex, and age would be the same, but like there are conditions that they like to match people in, and the more you're able to match the people, the stronger your results are, and so they kind of got away with a not particularly diverse population by doing that. There are other studies that have more diverse populations that corroborate these findings. Does that sort of help?
And
We'll just make our way across. Oh, that's just a shift.
Thank you so much, Erin. I just have a quick question about the previous slide about vitamin D, which I think maybe was asked in the Zoom chat. Or
Yeah.
okay, which was that you said that the conclusion of this is that they concluded that exposure to vitamin D before the age of 15 was a highly correlated risk factor for MS.
Yeah.
But you said that they were comparing people who moved to the UK before the age of 15, in which case they retained the level of likelihood of developing MS of India, Pakistan, Bangladesh.
Yeah.
Whereas people who moved after the age of 15 adopted the likelihood of British people. But
No,
I guess,
sorry, finish your
well,
thought.
I guess I don't know. I guess in both cases, both groups had exposure before the age of 15, and if anything, the ones who moved earlier have.
No, you're correct.
One
It's
of them.
it's the other way around.
Oh.
Oh, okay. I'm so sorry to be so confusing.
No, that's obviously the overall message.
Yeah. So it's exposure to vitamin D that is important when you are young,
Okay.
primarily. Yeah, I'm
Okay.
sorry about that.
Thank you. Thank you so much.
That's the problem with looking at so many studies.
It was the same.
Thanks, Erin, for that excellent presentation. I also had a question about related to the blood-brain barrier. So I was just curious, are there any theories out there for like how, like what the causal link is between having these specific risk factors and then the causal link between those and the breaking down of the blood-brain barrier? Like, are there any specific theories from like a biomechanical perspective and like how it actually might occur?
So in terms of, I'm not sure in terms of vitamin D, but in terms of smoking, we all know that smoking can cause damage to the cells in your body. So it's no surprise that it would also cause damage to the cells in the blood-brain barrier and break those down. In terms of the microbiome, we have what's called this gut-brain axis, which is this idea that bacteria in your gut can actually, they influence your whole body, but especially your brain. And so that could go into it a little bit, particularly factors that the bacteria are producing could be toxic to this blood-brain barrier. Epstein-Barr virus, as far as we're aware, does not infect the cells of the brain, but also it's a very sneaky and elusive virus. So it could be that it's there and we just can't detect it because it's hard to detect in blood. And in terms of stress, I guess it's involving the brain. You don't want to stress your brain out too much. So I think there's lots of things. When we induce it experimentally, we actually inject pertussis, which is it causes the symptoms of whooping cough. And so maybe that comes into it, but I wouldn't, don't go injecting yourself with pertussis is the take home from that. So yeah, if that helps.
Yeah, thanks.
So I have a question more related to I got curious about the Epstein-Barr virus. So usually the infection is very acute. So it comes and goes very quick, very fast, and it's quick. The thing is, later you mentioned that you have dormant viruses that they come back. How does it correlate with the acute infection? Like, are the same epitopes produced and later on?
So viruses have sort of two phases. They have a lytic phase, which is where they go in, guns blazing, destroy the place. And then they have a latent phase, which is they're like, I'm going to hide and I might reactivate later. And so, what I mean by that is they originally go in, they destroy the cells, and then they might think, oh, I'm destroying too many cells. I'll just kind of lie low. And it's that lying low it does it for the rest of your life. And it does it in the B cells specifically. And it could be that there are potential factors involved, such as these endogenous retroviruses that sort of reactivate this latent Epstein-Barr virus. But in terms of the different epitopes, they have different streams that they go in. So if they go to the lytic phase, they'll produce these proteins that are required to destroy the cell. And if they go the latent phase, they'll produce different proteins that help it sort of like embed into the DNA and things like that. Does that help?
Yeah,
Okay, cool.
that was clear. But do they manipulate the B cells as well?
Sorry?
Do they manipulate the B cells themselves to make them more prone to go somewhere or invade the brain?
That might be a direction. I'm not entirely sure, though, if they kind of like hijack it, so to speak, and alter its movement.
Thank you, Erin, for your very informative talk. My question kind of dovetails on the one before, and I was curious if you could talk a little bit about that last reactivation thing that you were talking about, that you're like and embedded in the genome, and also like if there's not just a correlation with Epstein-Barr, but if there's potentially like it's that B cell connection and there's actually something causative about it, or if like with the waking up.
No, I go in. Yeah. So up until now, a lot of our data has been correlative. So what people will do is they'll look at the blood of people with multiple sclerosis and they'll see if little parts of the endogenous retroviruses are being expressed. And say they see it more in people with MS than people without MS. They'll compare the two and go, oh, okay, this is more common in people with MS. But that's a correlation, and it's not actually looking at the action of these endogenous retroviruses and what they're doing. And that's what my PhD is looking at. So early days, but there are certain ideas that these endogenous retroviruses are potentially making the immune system produce some of these cytokines that we said earlier was like attacking the myelin sheath. There might be some where it's helping to activate the cell further. So EBV is already activating B cells. Imagine if an endogenous retrovirus comes along and like amplifies its activation tenfold. And so sort of potential mechanisms like that.
And maybe Zoom as well.
Yeah.
Thank you for your presentation, Erin. I always appreciate great graphics. My stepmom is living with MS, and so again, the lived experience of the people who are living with MS are so important to uplift in spaces like this. So I definitely appreciate you sharing and being vulnerable with that story. I have one clarification question. What type of virus, EBV?
What type of virus?
Yes.
Double-stranded DNA virus.
Okay, thank you. And then a follow-up question to that.
Herpes virus.
Yes.
It's a herpes virus.
And then a follow-up question to that. I study HIV, and there are a lot of viruses out there that do have a longer latent period, chickenpox, and we see that with shingles coming up in older populations. Is there a field of comparative virology about looking about how long these phases are and what types of viruses are in the body that have these symptomologies that come in later in life to see if there are any comparisons across different types of viruses and how they impact the body during these longer latent phases and then the reactivation?
Yeah, so to really think about HIV, it's a retrovirus, and what retroviruses like to do is they like to embed themselves in our genome. So the precursors of HIV are likely what are our endogenous retroviruses in our body now. And it could be in the future that HIV might become endogenized, although that's a few million years away, so your job is safe. And when we look at viral infections, it's the ones that cause maybe more of a latent infection that we see associated with these endogenous retroviruses. And then also what's interesting is sort of you have an initial infection and then autoimmune-like disorders. So HIV, there's one where it becomes more neurological and you get neurological conditions. EBV is linked to multiple sclerosis. There is a Coxsackie virus that is linked to development of type 1 diabetes. And then the one that we must all know about is COVID and long COVID. So if you look at the symptoms of long COVID, they're very autoimmune-esque. And that is an interesting part of research. And so actually what is one of my sort of big picture thoughts is linking up these viral infections to autoimmune diseases, because autoimmune diseases as a whole, we don't fully understand. We don't know what causes them. We have lots of like, oh, it could be this, it could be this, but we haven't been able to sort of round it out into a big picture of understanding like we have about cancer or infectious disease. And we have a lot more treatments for those type of diseases than we do autoimmune diseases because we understand them more. So if we understand them more, we have more treatments available. So yeah, thank you for your question.
Thank you.
Yes, I can go to
Oh,
the.
would you like to zoom in first and then beat you?
Ducky needs to ask a question. I don't.
Do you know how to get onto the Zoom?
Oh, yes. Let's give it a go.
Let Ducky speak.
Oh, chat. Okay. All right. Awesome. Okay, so I think we answered the thing about the vitamin D, and I can come forward to that. Okay, Jamila says, can you please say more about monoclonal antibodies and how they work for MS and other autoimmune inflammatory diseases? What are the benefits and side effects? So a monoclonal antibody works by binding to a particular protein that's expressed on the cell. So all the cells in your body have different proteins on the surface that sort of identify them. So just as we can tell that that's Riku because he's wearing glasses, they can tell that this is a cell because it has this marker on it. And we can get antibodies against various cell markers. So one that's been really successful has been ones against B cells, which is interesting because we previously thought that T cells were the ones doing all the attacking. And then we found that actually if we targeted these B cells with these antibodies, we had an improvement in MS symptoms. So when we think about benefits, the benefits are a reduction of relapses. Most of these drugs are designed for people with relapsing remitting MS. And so a lot of these drugs are designed at reducing the number of attacks and increasing the amount of time in between the attacks. But I lost my train of thought. So that's how they work. And then side effects. Side effects are generally, when you're messing with the immune system, you have the problem of immune suppression. And so you become more susceptible to various infections. And that's never something that you want. And also these treatments tend to be quite a lot. So imagine you've taken like ibuprofen, you've taken a painkiller, and you probably won't get a lot of side effects from that painkiller because it's quite a low dose. But when you get up to the really high doses that we're having to do for these antibodies, you can get general ickiness, so headache, fever, fatigue, that just makes you feel ill, and can make you feel like the treatment isn't worth it. Another thing is if we have to give these treatments intravenously, these individuals have to go to hospital and get it done. They can't just pop a pill at home. I hope that answers your question, Jamila. Ducky said, I read about some promising preclinical studies which found remyelination. Can you talk about that for a moment? Yeah, so remyelination is where we have, so I talked previously about the myelin sheath and how it is all the way back here and how it gets broken down by immune cells. And there are other cells in the brain that are responsible for remyelinating and for making the cells that help to form this myelin sheath. And there are some drugs that are aimed at activating these cells more, so they produce more protein that can be used to make more myelin sheath. Or that again, that's also more helpful at the beginning of the disease course, because as the disease progresses, the cells that are responsible for remyelination start to die off because the individuals are getting older, but then are also potentially involved in this immune storm being created by these immune cells. And so it, like, the drug that allows for remyelination is awesome, but again, I think it's more of an early treatment. And it's hard to have more drugs that are helpful towards the end of the disease course. Craig says, is there a connection between diabetes and MS? There is in terms of the genetics. So if you look at the genetics of people with diabetes and people with MS, they're very similar genetics. And I believe they can also co-occur at a higher rate than in the general population. I think especially as the individuals get older. Now, again, I'm not sure if it's type 1 or type 2 or a mix of the both. I also know that if you have family members with multiple autoimmune diseases, such as myself, on both sides, you're more at risk of developing autoimmune disease. You know, I've got to 28, so we're good. So people who move from India, Pakistan before the age of 15 get the British risk, and people who move after 15 keep the risk. So it's not exposure after age 15. So in India and Pakistan, they have higher rates of sunlight and vitamin D. And so that the more vitamin D you have, the lower your risk of MS. And it's particularly when you're younger. I hope that's helpful. I can send you the paper if you want to read it. There are a huge number of diseases which have long diseases. Cool. Okay. Do the maths which target B cells, target all the B cells or a subset of the B cells. The ones I'm thinking about target all, well, the majority of the B cells, but you can target some of the B cells by looking for these markers that are on certain B cell subsets so you're interested and not on the others. So it's only going to target the ones you're interested in. But I think that is the end of, has anyone studied incidence of MS on ancestral groups in higher latitudes? Interesting. The idea being that at the higher latitudes, you'd have more access to vitamin D. Less. Oh, higher latitudes, sorry, I was thinking altitudes. So there are actually, so there's been research that obviously when we look at ancestral groups, there is historically a lot of hesitancy to be involved in research studies because these individuals and these groups have been exploited in the past, and that's fair enough. When we are able to look at these individuals, we actually do see a higher rate of MS, particularly in the Indigenous peoples of Canada, which are interesting to study because they're at that higher latitude, especially in, say, the Yukon, where there's not a lot of, like, you have polar night, polar, like, darkness during the ones at the night. Are these for those who follow traditional diets? So there's also a lot of research that goes into traditional diets and development of MS, and that is actually something that people are researching at UBC. We have some individuals who are basically looking at different diets that people consume and their risk of MS. Okay, one more question. We'll take it from the audience.
Ducky needs to ask more questions. My question had to do with the environmental factor of microbiome, and if that has anything to do with epigenetic connections, and if there's therapies that have to do with epigenetics, research in that direction. That's kind of like where I was the last place I left biology, so I'm really curious.
Okay, so I'll explain epigenetics for those who might not be up to date on it. So epigenetics is this idea that, yes, your genome is fixed, but we can change the way that it is expressed by your environment. And so you're talking about specifically the microbiome. And so we have this gut-brain axis, and everything that your gut creates in the microbiome can affect the way that certain genes are expressed throughout your body, including your brain. So I'm sure there are epigenetics involved. I don't specifically look at that side of epigenetics as much, but it is something that is quite interesting, especially when you look at the reactivation of something. So, yeah. Okay. That's all we have time for.
That's good chat, that's all.
Oh, yeah.